Okay. It's almost starting. So I think I'm going to admit everybody now.
So then you'll have to do that again after a while, I guess. Right?
Yeah, okay, I'll wait another minute then.
No, you can start now, but then you'll have to do it again because some people will be late, I suppose.
Yes. Okay. So I think I'm going to admit everybody now. Oops.
So you'll have to do that again after a while, I guess.
Yeah, okay, I'll wait another minute then.
No, you can start
That's
now.
the YouTube live stream, Gilad. You need to stop it on your.
Yes.
Okay.
Okay. Okay. I'm going to start the recording as well.
Hi, Sheena.
Hi, Jane. How nice to see you.
Lovely to see you.
Hello.
Oh, wow.
Hi,
so
Sheena.
many
Hi,
people.
everybody.
Hi, it's nice to see some faces. Miss you all.
We miss you too.
Good to see you.
Lila, hi. Hey, oops, I
Great to
maybe
see you,
muted.
everyone.
Trisha. Susan.
Hello, Sheena.
Hi.
Hi, Sheena.
Oh,
Everybody.
hi. Nice to see you. Wow.
Hi, everyone from Stockholm.
Lovely to
Gunnar.
see you. Hi.
Hi, Gunnar.
Hi everyone, this is Tarak from Switzerland, Lugano.
Ah,
Nice to meet you all.
amazing.
Sorry.
Hi, Jane. Hello, Sheena.
Hi.
Arizona.
Hello.
Hi, Sheena.
Hi. Everyone's backgrounds look much more interesting than mine.
Hi, everybody.
Oh, hi. Nice to see you.
Oh, here's Ken. Okay.
Oh, hello, Gilad.
Hello.
So the numbers are still increasing, so let's wait another minute, maybe.
Yes.
Hello from Argentina.
Hi.
Hello.
Hi, Sheena.
Lovely to see you.
Good to see you too.
So it's 1 past the hour, and since I'm doing this from Switzerland, maybe we should start. And so it's wonderful to see so many of you again. Welcome to our webinar today in our series of webinars on protein folding and dynamics. Following the huge interest of last week or 2 weeks ago, we have now increased the maximum number of participants to 1,000. So we hope that everybody is able to join. In case you're not able to join, there's also a live stream on YouTube that you can either watch during the seminar or later on. And you can see this. So let me now share my screen with the announcement of today's seminar. So I hope you can see my screen. And so you will find the current talks that we have planned on our website. www.proteinfoldinganddynamics.com. You will also find the link to the YouTube channel there. And it still has a funny long name, but as soon as we have more than 100 subscribers, we can choose a proper name. So please do go and subscribe. And so there you will be able to watch all of the talks of this series also later on. And so our lecturer today is Sheena Radford, and it's wonderful to have her here for our next webinar. And so most of you know Sheena, so I don't have to introduce her in great detail, but for the 2 or 3 of you who may not know Sheena, let me just say that she is the Asbury Professor of Biophysics in Leeds. And among other distinctions, she's a Fellow of the Royal Society. She's a Fellow of the Academy of Medical Sciences. And she runs an extremely versatile research program using a broad range of biophysical methods. And her topics are protein folding, aggregation, and amyloid diseases. And her talk today will be about amyloid assembly, the Achilles heel of a disease mechanism. And so I would like to ask all of you except Sheena now to mute your microphones so that we can listen to this without disturbing background noises. And then there will be the opportunity to ask Sheena questions after the talk. To do that, please use the chat window and either raise your blue digital hand or just write into the chat, and then we can call you and then you can ask your question unmuting your microphone. And so with that, I don't want to steal more time from Sheena, so I will stop sharing. And so I will ask Sheena to share her screen, and we're very much looking forward to your seminar. Sheena.
So just to say hi to everybody, it's really great. Oh, I turned myself off so I can't see myself. It's really great that you've all joined in to listen to what I have to say today about early steps in amyloid assembly. But of course, I really want to start by thanking Gilad and Ben and Hagen for setting up this webinar so that we can see each other from around the world when we're in different stages now, I understand, for lockdown. So I listened to Jane's talk on the internet the other day, and she talked very nicely, I think, about how intrinsically disordered proteins can undergo conformational changes that depend on the solution conditions and the partner proteins that they bind to. And I'm going to follow up on that kind of thought today, but we're going to look at the protein-protein interactions that drive assembly into these beautiful, highly ordered amyloid fibers. And specifically, I'm going to look at the early steps in amyloid assembly and to see if we can find the Achilles heel in our disease mechanism. So, why can't I change slides? Great. So without further ado, I think I really want to thank the people in my group who have contributed to the work I'm going to talk about today. So many members of my group, so the current amyloid team, are shown here. And for the talk that I'm going to give today, there's going to be a green team and a red team. So the green team are the beta-2 microglobulin team, and the work I'm going to describe is Theo, who's now left the group. He's at the NIH. Emma Kaywood, who's there with the green arrow, she just was leaving the group when this photo was taken last year, with Matt, Arnout, Gary, and my academic collaborators, Andy Wilson and Eric Hewitt. And then the red team, that's an alpha-synuclein team. That work was done by Sabina when she's still in the lab, and Roberto here, still in the lab, also done with Kieran, who's now in industry. And thanks to all of you that contributed to this work. And my golly, seeing your pictures really makes me miss you. For those of you around the world, we in Leeds are still in 100% lockdown. So it's 8 weeks since we've been able to see each other in the flesh. So with that emotion, let's move on to do some science. So I think everybody listening to this talk will know about amyloid fibrils and the cross-beta structure. And this introductory slide is just to share with you that there are 50 known human proteins that form amyloid fibers that deposit in different parts of the body and give rise to different amyloid diseases. And as I mentioned, I'm going to give two stories today: amyloid aggregation for beta-2 microglobulin that gives rise to dialysis-related amyloidosis, and aggregation of synuclein involved in Parkinson's disease. So to summarize a massive field in just a couple of points, I think the key points here are that all amyloid fibers have a common cross-beta architecture, and we're now seeing beautiful amyloid architectures from solid-state NMR and cryo-EM data. And I'm not going to talk about that today, although we do solve structures of amyloid in my lab with Neil Ranson and his colleagues. One of the key questions then is, so how can so many different proteins, that whether they're peptides that are unstructured or intrinsically disordered proteins, or structured proteins like immunoglobulin light chains or beta-2 microglobulin, how can these different proteins form this common cross-beta structure of amyloid? And why do they deposit in different organs and give rise to different diseases? So even if the protein is found systemically, amyloid deposition tends to be rather specific. And of course, what we don't understand in the field is what causes amyloid disease. So we don't understand the mechanisms of disease, and the strategy of who to treat and when to treat and how to treat individuals remains a big mystery. And of course, as we'll all be aware, these diseases are found throughout the human race. And so a major question is who to treat, how to treat, and when to treat individuals. So from a structural molecular viewpoint, which I think people listening in today will be mostly interested, what we want to understand is the structural mechanism of amyloid formation. That is, how is the native precursor of a protein, native state of a protein, what conformational changes does it undertake to initiate amyloid formation? What's going on with this slew of oligomers that form during the lag phase of assembly, and many are thought to be involved in toxicity, again by unknown mechanisms? How does the conformational change to the cross-beta structure of an amyloid fibril occur, and that then form these large plaques? So we want to understand the structural molecular mechanism of amyloid formation. We want to understand the structure, the conformational properties of every species on this reaction coordinate. And we want to put numbers on these arrows. So we want to understand the rates of interconversions and the populations of different species as a function of time, as a function of environment. And only when we can map the population and the identity of these different species can we start to think about which species are cytotoxic, and maybe to then inspire disease prevention, amelioration or treatment. So that's the big question. And of course, the challenge is we're having to deal with rare events, rare conformational states, and dynamic ensembles, the kind of thing that is challenge for health, but fascinating for this audience. So I'm going to focus today on these initiating stages of amyloid formation, where the protein will start to misfold as a monomer and form the first protein-protein interactions that commit a protein to an amyloid pathway, which thermodynamically, of course, is a downhill process. And so I specifically want to ask the question about how specific are these early protein-protein interactions in amyloid assembly. And if we can map those key initiating amyloid interactions, if there are any, can we target these interactions to ameliorate amyloid disease? So I'm going to exemplify these two big questions with two example proteins, as I mentioned. The first is beta-2 microglobulin, and we chose this as an exemplar of a protein that starts its life with a nice natively folded structure and has to unfold or partially unfold and reorganize into the cross-beta structure of amyloid. And beta-2 microglobulin deposits into amyloid fibers in the joints of patients undergoing with kidney dysfunction, so that it's called results in a disorder called dialysis-related amyloidosis, as you can see in this scintigraph here. And the second example is kind of the other end of the other side of the same coin. What happens when amyloid is initiated from an intrinsically disordered protein? And here we've chosen alpha-synuclein as our exemplar. And you'll all know that alpha-synuclein deposits in amyloid fibers in Lewy bodies in the substantia nigra and gives rise to the disorder Parkinson's disease. Okay, so let's start the first story then. This is beta-2 microglobulin. Beta-2 microglobulin is the light chain non-covalently bound to the heavy chain in the MHC class 1 complex that's displayed on the surface of all your nucleated cells, displays peptide antigens, and is essential for your immune response. Now, what you might not be aware of is the catabolic cycle of beta-2 microglobulin is it folds, but then it dissociates from the MHC heavy chain and is normally degraded in the kidneys. If you have renal dysfunction, you can no longer do this. And beta-2 microglobulin, the concentration in the serum increases about 50-fold. And classic cross-beta amyloid fibers then result, I've already shown you this image, that deposit specifically in the joints and give rise to pathological bone destruction. And with Neil Ranson and Matt Iadanza with using cryo-EM and Bob Griffin using solid-state NMR, we recently solved the structure, at least of the wild-type protein in amyloid fibers. So let's look now at our precursor protein. This is beta-2 microglobulin, a classic immunoglobulin fold with 7 antiparallel beta strands. What you also need to understand here is that the key points of the protein is that the N-terminal 6 residues of the protein are clipped off in about 1/3 of the protein in amyloid fibers, causing the production of this protein I'm going to call delta N6. Now, this N-terminal 6 residues juxtaposes with a proline that stabilized in a normally unfavorable cis conformation gives a nice hydrophobic patch to the top of the protein here that's functionally required to bind to the MHC antigen. So the beginning of this story then came from Tino Eichner and Theo Karamanos, both when they were PhD students in my lab, and we're trying to grow amyloid fibers from these two proteins, the wild-type protein and delta N6 in vitro in the lab. And you can see whilst the wild-type protein is rather, doesn't like forming amyloid fibrils in vitro at least, as measured by thiot fluorescence and negative stain EM. If we just simply clip the N-terminal 6 residues off the protein, it rapidly aggregates into very nice amyloid fibers. Now, Timo was able to solve the structure of beta-2 microglobulin, both the wild type and delta N6 using NMR, and I showed those solution structures here. Now, what you can see for delta N6 is the protein retains its native all antiparallel immunoglobulin folds. But what you can't see is this protein is virtually not protected from hydrogen exchange. It's really dynamic. So whenever I show you a figure of delta N6, I want you to think about it as a highly fluctuating structure that nonetheless retains this overall immunoglobulin architecture. Now, removal of this N-terminal 6 residues has another subtle but important conformational consequence, and that is that this cis-proline flips to form a trans-proline and becomes buried in a new hydrophobic core that has other structural rearrangements. So the take-home message for this introductory slide, therefore, is that isomerization of a single peptide bond around proline 32 is sufficient to switch on amyloid formation. So going back even further than that, Thomas Jahn, when he was in my lab over 14 years ago now, was doing classical protein folding and unfolding studies of human beta-2 microglobulin. And as you'd expect from a protein that has a cis-proline, it folds via a long-lived kinetic intermediate with a trans-proline 32 that we call I-trans. And by comparing the chemical shifts of delta N6 with those of the I-trans species captured in real time by NMR, we're able to show that the kinetic intermediate I-trans and the equilibrium intermediate delta and 6, have superposable structures. So delta N6 is a kinetic mimic of the transient folding intermediate. And this species, the concentration of I trans could be related directly to the rate of amyloid formation. And this allowed Thomas at the time to show that aggregation of delta N6 or aggregation is directly dependent on the concentration of this I trans species. So this gives us the first hint that aggregation is specific because this precursor is necessary and sufficient to drive aggregation. And Theo was also able to look at an earlier intermediate on the folding reaction coordinate that's more molten globule-like. Its N-terminal 20 residues are now intrinsically disordered. It has no chemical shift dispersion and broad lines. And this species is not able to form amyloid. So the take home here is amyloid is initiated through this very specific I trans or delta N6 state. We also know that the amyloid fibers by solid state NMR with Bob Griffin have a canonical parallel in register structure. So we have to transfer an anti-parallel monomer into a parallel beta stranded amyloid fiber. What I want to talk about today, more than these initiating and final species of amyloid formation, are these early intermediates, and ask the question I posed a while ago: how specific are these interactions, or are these interactions non-evolved, non-specific stickiness between partially folded non-native states? When does a conformational transition to cross beta occur? And if we can map these interaction interfaces, can we develop inhibitors that target these early protein-protein interactions? So to do that, coming back to this original schema I've shown you, it's clear that what we need to do is combine a lot of kinetics that allow us to map the course of aggregation and some biochemistry to identify the types of oligomers that are formed during aggregation. But then to transplant on that kinetic view, structural information, the only approach you can use for these dynamic and interconverting species is dynamic experiments using NMR. And this is what Theo did. So he used a whole variety of kinetics and NMR experiments to map the entire energy landscape and structural landscape of aggregation. And that's what I'm going to tell you now. So to start off then, Theo was doing some kinetics, some classic seeded fibril growth kinetics, where we take delta and 6 amyloid fibers grown in vitro, feed them with delta and 6 monomers, and measure the initial rate of fibril growth. And so that initial rate versus protein concentration is shown here. And what this shows you is clear non-linearity. And that's very unusual behavior. Normally you see a linear dependence of the aggregation rate on concentration consistent with monomer addition required for fibril growth. So what this bifurcation tells us is clearly that one or more higher molecular weight oligomers are essential to add on to fibril ends to grow amyloid fibers. And the big question is what's the identity of these key oligomeric species? So Theo did a whole variety of experiments: native mass spectrometry, size exclusion chromatography. But I just want to share with you now sedimentation velocity analytical autocentrifugation results of the beta-2 microglobulin as a function of protein concentration. And I thought for this, what Theo would see as a whole Himalayas of different molecular weight species, akin to this slew of oligomers that form normally during amyloid assembly. But we saw the opposite. We saw very clear, highly populated states as a function of protein concentration, consistent with monomer, dimer, and a species probably hexameric in structure, with little species of intermediate molecular weight. And so we can fit this equilibrium to a 1, 2, 6 monomer dimer hexamer equilibrium. Now, if we know that, we can look at our protein by heteronuclear NMR. Here I'm just showing a zoom in of some of the chemical shifts, and we can watch these chemical shifts changing as we change the concentration of protein in the NMR tube. And we can plot these differences in chemical shift as a function of concentration and fit this equilibrium to a 1-2-6 reaction. And this allowed Theo to estimate that the dimerization affinity is low, it's about 50 micromolar. But the hexamers, we can fit these data to a model in which the hexamers form by the trimerization of dimers, so 3 dimers coming together, and this is remarkably tight. So it's a 1-2-6 equilibrium. So now we know the species that are present. So of course, the next key question is, what's the structure of these dimers and hexamers? Is there one specific structure, or are there many dimeric and hexameric conformations present in these experiments? And so now we have to move to our NMR data to obtain some structural restraints. Now, fortunately, there's a whole slew of NMR experiments being developed that Theo very nicely reviewed in this review a few years ago now, that allow us to look at conformational changes between unfolded and folded states, for example, to map transient interactions between monomers and dimers and oligomers. And these are CPMG or relaxation dispersion experiments. Paramagnetic relaxation enhancement experiments, you can do NOEs, and I've already mentioned real-time experiments, real-time NMR experiments. And Theo did a lot of experiments using CPMG and NMR PREs. And just to fill out this reaction coordinates, a whole array of very beautiful NMR experiments developed by Lewis Kay, Marius Klor, and others that even allow us to look at interactions between monomers, oligomers, and higher molecular weight species like fibrils. So NMR is ideal for mapping these early protein-protein interactions, and Theo did a huge array of experiments. Now, I'm not going to show you any of the NMR data at all. I just want to jump to Theo's final structural model. And that's shown in this very pretty picture here. And we just published this at the end of 2019 in eLife. So anybody wants to look up some of these modeling and NMR experiments, you can talk to Theo or look up this paper here. And if desperate, you can even talk to me. So what did we find in our filling in our reaction coordinate then? So what Theo was able to show is these early dimers that form during amyloid assembly have a specific interface, and it forms this very long, elongated head-to-head structure. 3 of these dimers then curl round each other to form a beautiful, compact hexameric structure. And then we think that the free energy of binding of the dimers together to form the hexamer unfurls the C-terminal beta strand of the protein. We could see that in our NMR chemical shift data. And somehow this allows the protein to add onto the end of a fibril seed. And somehow, by a mysterious yet to be understood mechanism, this allows conformational change to a parallel in register structure. So clearly, the end of this reaction coordinate, we still have some work to do to understand where the conformational conversion actually occurs. Now, in his PhD work in my lab, Theo was also able to map the structure using NMR of a dimer formed between delta N6 and the murine beta-2 microglobulin. Mouse beta-2 microglobulin is 70% identical and 90% sequence similarity to the human protein. But interestingly, the murine protein is not amyloidogenic, even if you unfold the chain. And if you mix the mouse protein with a human delta N6, you can arrest fibril assembly. And Theo was able to map the structural properties of this inhibitory dimer. So what I want to do now is look in more detail at this productive on-pathway dimer and the inhibitory dimer. And they're zoomed in here so you can see them. So here we have the elongated on-pathway dimer. You can see it's a head-to-head interaction. All 3 loops at the top of the protein with proline 32 at its epicenter form the nature of this protein-protein interaction interface. By contrast, in the inhibitory dimer, again, you can see it's a head-to-head interaction, but the angle between the monomers is much more acute, such that only two of the three loops are at the interaction interface.
Sorry,
Again,
Sheena, let me just briefly say, could please all of you switch off their microphones? Thank you.
so what do we take home? Thanks, Ben. So what do we take home from this? So what we learn from this is that weak binding is sufficient to either drive or inhibit aggregation, giving hope for making inhibitors because we haven't got to inhibit very tight protein-protein interactions. It also tells us that the nature of these first interactions is very specific and determines whether a dimer is productive or inhibitory. And of course, the fact that these interaction interfaces are specific gives us hope that we can target these interaction interfaces with small molecules or other methods to think about inhibitor design. So with that challenge of inhibiting interaction interfaces to prevent amyloid assembly, we set Emma Kaywood, who you saw on the first slide, this is her thesis work, we set her the enormous challenge of seeing if she could find small molecules that would bind to delta N6 and prevent its assembly. Now, this was an amazingly difficult challenge. Remember, delta N6 is this partially structured, highly dynamic state. It's not a native static structure. And that bestiary microglobulin doesn't bind any ligands. It has very flat surfaces and no obvious binding pockets. So Emma's thesis, which was done with Professor Andy Wilson in the School of Chemistry and in the Asprey Centre, Emma used computational methods and mapped two patches or two targets. She called it site 1 and site 2. On each side, if you like, of the beta sandwich. And here is the BC loop close to proline 32 that will be up here in both structures. So she mapped two sites, one on each side of the molecule, that she thought by computational methods over all the structures in the NMR ensemble may have good ligand binding properties. And then she cleverly, what I thought was cleverly, used this wonderful technique called disulphide tethering to search for small molecule ligands that would bind to this interface. And then because the ligand library she made all had a disulphide on, we'll be able to form a disulphide bond to S33 that we will mutate to a cysteine, or to serine 52 that we mutated to a cysteine, or to, on the other site, L65 that she mutated to a cysteine. And so this experiment works because the ligands that bind most tightly form the most disulphide bonds to the tether that you put in, and you can pull out the most tight binding molecules by quantifying covalent modifications using mass spectrometric methods. So I just want to show you one result from Emma's thesis as a proof of principle. So here I'm showing you binding of these two ligands, beta-mercaptoethanol, which was our control molecule, and one of the ligands from Emma's library called S54 that we are going to covalently modify in serine 52 that targets this site. And we're measuring the rate of aggregation of delta N6 covalently modified with each of these ligands. And what I want you to see from these data is that while putting a mercaptoethanol moiety on serine 52 has no effect on the initial rate of aggregation, if Emma puts this molecule, this small molecule 54 on this residue 52, she can really slow down, if not almost completely ablate, aggregation of delta N6. So the take-home message from this is that this provides proof of principle that we can inhibit aggregation by targeting those specific early interaction interfaces. And a second important take-home, as it shows that these interfaces are both necessary and sufficient to drive aggregation. So what's the take home on this B2 microglobulin story then is, I think there's a remarkable specificity of the protein-protein interactions that initiate amyloid formation, and that through NMR and other methods and combining them, we have the protocols we need to map these interaction interfaces, and that targeting these early interaction interfaces, even in these non-native molecules, can be used to inhibit amyloid formation. So we learned all this about a molecule that starts from a native structure. Now I'd like to sort of ask, how relevant are these kind of thoughts for aggregation mechanisms that start from an intrinsically disordered protein? And the one we chose to study is the 140 residue alpha-synuclein. And as I mentioned, this work was done by Sabina and Kieran and Roberto. So a little bit about alpha-synuclein before we get into the aggregation. So it's 140 residue IDP. The N-terminus is amphipathic and has these KXK-EGV motifs. And this is where all the familial mutations that cause Parkinson's disease map to. In the middle of the sequence between residues 60 and 90, there's this so-called NAC region, which is highly aggregation prone and known to be necessary and sufficient to drive aggregation of full-length protein into amyloid fibers. And the C-terminal region has a lot of acidic regions, so it's highly acidic. Now, alpha-synuclein is known to form alpha helices upon membrane binding that span the N-terminal region and through the NAC region. And this is thought to be important in the function of alpha-synuclein in membrane remodeling and vesicle recycling at the synapse. So we wanted to look at the early protein-protein interactions that modulate aggregation potentially of this IDP. So to get a hold of this, Kieran, as a PhD student in David Brockwell's lab, used dynamic force spectroscopy. And I think this is a neat way of looking at protein-protein interactions, because in a single molecule experiment like this, you only look at those first dimerization events. We can't form any higher order species. So we covalently attach molecule of interest, synuclein in this case, to the cantilever and the substrate. And if they interact and form a force-resistant interaction interface, and we pull the cantilever away from the surface in a retraction, we will measure a rupture force that tells us how strong the interaction is, and a rupture distance that tells us potentially where the interaction interface is. Now, if the interaction is specific, that rupture will have a defined force at a defined distance and will give us an interaction hotspot. If there's no interaction or weak interaction, we won't see anything. And if there's a non-specific interaction, we'll have very broad peaks and no hotspot. So Kieran went away and immobilized alpha-synuclein at its C-terminus and carried out these dynamic force spectroscopy experiments. And much to my astonishment, he saw these beautiful rupture peaks consistent with a strong interaction between these IDPs. When we put those data onto these 2D hotspot plots, we can see we see a very nice hotspot with a strong force, about 50 peak newtons, a retraction speed at 100 nanometers per second, and at a specific distance of 30 nanometers. So this suggests that these monomers interact via a specific force-resistant interface. And the question is, where is that interacting surface, at least as detected by this experiment? Well, you can get at that by measuring the distances that the rupture force occurs over. So in this example, if the interaction interface is in the middle of the protein, you will disrupt that interaction at a shorter contour length than if the interaction is at the termini of the protein, as shown schematically here. We can also get at it by mobilizing the protein at different places via cysteine and measuring how the immobilization point changes the distance to rupture. So in this case, if the interaction interface is near the C-terminus and we immobilize at the C-terminus, the contour length at rupture will be shorter than in the alternative scenario shown here. So summarizing this work from Kieran, what this allowed us to point to was that this first interaction interface occurs roughly between residues 40 to 60, a region, interestingly, which contains these familial point mutations and juxtaposes the NAC region. And so this force is also, high force is also consistent with the interactions between beta strands. So we started looking in detail at this region 40 to 60. What Kieran and Sabina noticed when they looked at the sequence of alpha-synuclein over its 140 residues using algorithms predicting amyloid propensity or solubility, or steric zippers using David Eisenberg's algorithm, you can spot the NAC region known to be necessary and sufficient for aggregation. But you can also spot these two other regions clearly in the region 40 to 60 that we'd highlighted. These regions, which we call P1 or P2, have a high amyloid propensity, low solubility, and P2, but not P1, is predicted by David Eisenberg's algorithm to form a steric zipper. And in fact, David already shown very beautifully that peptides equivalent to P2 are able to self-assemble into amyloid-like microcrystals in vitro. So Kieran and Sabina set out to have a look what P1 and the effect of these regions P1 and P2 have on aggregation of full-length protein by deleting the sequences from P1, P2, or both sequences. And I'm just going to summarize their work grouped into three areas in the last minutes that I have. So we're going to look at aggregation kinetics judged by a thioflavin T fluorescence, conformational properties with a brief look at some NMR experiments, and then some functional properties. So let's look at aggregation. Here is alpha-synuclein aggregating at neutral pH slowly and more rapidly at acidic pH. If we delete P2, nothing happens. The rates of assembly aren't changed very much and fibrils result. The striking result I'd like to share with you is this one, and we were stunned again by this result, that if we remove the 7 residues equivalent to P1 in this 140 residue protein, we can completely stop aggregation over this timescale in our experiments, and at acidic pH, aggregation is slowed. And if we delete both of these regions, alpha-synuclein doesn't aggregate anymore on this timescale. So removing just 7 amino acids in P1 is enough to completely inhibit aggregation at neutral pH, whereas both regions P1 and P2 are needed to inhibit aggregation at acidic pH. Is this just an in vitro phenomenon? So we grouped up with Patricia Van Oosten-Hall, Sarah Good, and our master student at the time, Gemma Makepeace, and they put wild-type synuclein delta P1 or delta delta into C. elegans and looked for amyloid aggregates in the body wall muscles using a YFP construct. And again, what I thought was pretty exciting and stunning is if you remove just 7 residues in delta P1 or both delta 1 and delta P2, we see no aggregation in vivo, and using other phenotypic assays, the worms wiggle away happily, doing all the things worms are supposed to do. So P1 is required, just this 7 residue sequence is required for aggregation in vivo. So knowing that P1 is so important and P2, what do these regions do to the conformational properties of this IDP? And so what Sabina and Kieran did with Roberto Meyer-Martinez, our resident NMR expert in the group, is they used NMR PREs to look at the conformational properties of the synuclein. And so they put an MTSL spin label either in the N-terminus of the protein or in the NAC region or in the C-terminus of the protein and measured interactions across the IDP. And what you can see here is, has been shown by others before us, that the N-terminus, the basic region of alpha-synuclein, does interact with the C-terminus, making this IDP very compact. That was known very well from Marcus Fextetter and other people's groups, Thomas Knowles and Michaela Vindruscolo. But under a low salt and low pH conditions, hopefully you can see in these plots, we can see a very specific interaction of the P1 and P2 regions, both with the N-terminus, the NAC, and the C-terminal region of the protein. If we change the conditions to slow aggregation, the strength of these intramolecular PREs is reduced. And so we call these P1 and P2 regions master controllers of assembly. They're driving aggregation of the molecule, controlling aggregation by controlling the conformational properties of the IDP. So finally, in the last data slide, what do P1 and P2 do to the function of synuclein? Why does synuclein have these regions that make it aggregate? That makes no biological sense. But of course, we know the N-terminal region of synuclein is functionally important. So what role do these regions have in function? So following some very nice data from Gabby Kaminsky's lab in Cambridge in the UK, we added wild-type synuclein to DMPS liposomes, and we're able to show that wild-type synuclein binds to liposomes with micromolar, submicromolar affinity, and has already been shown in the literature remodels these liposomes to form lipid tubules. Interestingly, however, if we look at the delta-delta variant, the variant still binds to liposomes, it's about 10-fold weaker, but this function of the molecule to cause lipid remodeling into tubules can no longer occur, and the liposomes actually undergo fission and give us lipid debris, as seen here in negative stain EM. So I think what we learned from today's talk is that for synuclein, aggregation can also be highly specific in that these regions that abut the most aggregation prone region, the NAC region, form these specific but transient intramolecular interactions in the IDP that are required for aggregation to occur. They form the first intermolecular protein-protein interactions that drive amyloid formation and form this dimer that is force resistant, as I showed you by dynamic force spectroscopy, but are functionally important for membrane modeling. So what's bad for aggregation is good for function, showing this conflict between the need to evolve protein sequences for functional reasons that have net negative consequence for aggregation. And then finally, Wolfgang Heuer and Alex Buell had published a paper when we were doing this work looking at alpha-synuclein bound to one of their nanobodies, which they call a beta-rapin. And what's utterly fascinating is they found their beta-rapin that prevents both primary and secondary nucleation in their hands. And then they solved a solution structure of alpha-synuclein bound to their beta-rapin, showed a lovely beta-hairpin structure identical to our P1 and P2 regions. And then more recently, Sebastian Hiller with Bjorn Burman in Nature had shown that the N-terminal region of synuclein is canonically used to bind a whole slew of different molecular chaperones. And we can paint another dimension to the inhibitory effect of these molecular chaperones on aggregation in that they bind to our master controller region and stop these regions from driving aggregation. So to summarize then this whole story, what we've learned about these early protein-protein interactions in amyloid formation using both beta-2 microglobulin and with alpha-synuclein, I think what we've learned is that these early stages in aggregation are much more specific than we had originally, or at least I had originally thought, and that by mapping these early protein-protein interactions in amyloid assembly gives us a site and an opportunity to intervene in disease by targeting these specific yet weak and important early protein-protein interactions, suggesting that perhaps really they are the Achilles heel of a disease mechanism. So that's all I wanted to share in the time I have with you today. I just wanted to finish by thanking the funders, without which, of course, we couldn't do anything. This is a picture of the whole of the joint Radford and Brockwell team. We share a lab, and I really miss you all. We're in week now. We're still under total lockdown, at least in the north of the UK. I can't wait to see you all again and to do some more science. And I'm now very happy to take questions, and I'll stop sharing my screen.
Thank you very much, Sheena. Maybe you can leave the screen up, then maybe we can go back to individual slides if needed. So the first questions are already in the chat. If you want to ask questions, either put them in the chat or put up your, raise your digital hand in the list of participants. And so we can just start looking at the questions here. And so I will ask you to ask your question directly if you like, otherwise I can read it. So the first question is from Siddhartha Banerjee. Would you like to ask your question directly? Then please just unmute yourself and ask the question.
Yeah, thanks. Hi, Sheena. It's a wonderful talk. It's always wonderful to hear from you.
Thank you.
So the striking difference you showed, like the del N6 for beta 2 globulin. And so I was wondering, is there any known mechanism which is in place that chops that protein, that 6 residues, or you just wanted to test it with 6 residues of aggregation?
So as I mentioned, the original inspiration for that was that a third of the protein found in vivo in amyloid deposits of beta-2M does have that 6 residues clipped off. And it's clipped off by a matrix metalloproteinase. We think. We tried to map specifically which matrix metalloproteinase it was, but I think it's a non-specific cleavage at a lysine residue. So I think that the N-terminus is dynamic and is probably non-specifically clipped off by a proteinase, and then that sort of initiates the reaction, amyloid reaction, to occur by all of the features that I've shown you today. So I think it's a non-specific cleavage with a general lysine specific proteinase.
Okay, thank you.
The next question is from Sayanta Mahapatra. Would you like to ask your question yourself?
Yeah, hello.
Hi.
Hello. Hi, Sheena, such a nice talk.
Thank you.
Yeah, so my question is, in case of beta 2 microglobulin, you said that first there is a formation of this hexamer which interacts with the seeds, and then it matures into the higher order amyloids. So my question is how the scenario is different in case of non-seeded aggregations. Though I know that it's very infrequent event in cellular context, but my question is how the non-seeded aggregation or conformational conversion is different from a seeded in case of amyloid proteins.
That's a really good question, and one that I can't answer definitively. I think we assumed that the correctly or incorrectly that the non-seeded and seeded aggregation mechanisms would occur by a similar mechanism, because the hexamer forms in solution without the seeds and then adds on to the seed ends and catalyzes the reaction. We've tried over the years to measure, to follow aggregation of delta and 6 by measuring the concentration dependence of the rates of aggregation, thereby to pull out a kinetic mechanism. But it turns out that it's really complicated because the concentration dependence is non-trivial, let's put it that way. So it was very hard to definitively show kinetically that the unseeded and seeded reactions are the same mechanism.
Oh, thanks a lot. Thanks a lot.
A good question. I don't know the answer.
Yeah, thanks.
The next question is from E. Martin 1. Maybe Eric Martin, would you like to ask your question?
Yeah, this is Eric Martin. I was curious about the pH sensitivity with respect to the P1 and P2 regions of alpha-synuclei. I was wondering if you thought that these two regions contained or were responsible for the pH sensing, or if this was just a completely separate process and P1 and P2 were required for some early stage of aggregation and the pH is maybe irrelevant to that.
Yeah, so that's a good question. So we chose, I should have said this, we chose neutral pH and pH 4.5 because synuclein is known to spend part of its life in the cytoplasm and part of its life in other acidic organelles like lysosomes. So they were chosen to be kind of physiologically relevant, and you can choose which pH you think is the most relevant. It's very hard to unpick anything to do with the pH dependence of synuclein aggregation, given that it's full of acidic residues, all of which are titrating. Right. However, the P1 has no titratable residues in it. P2 has a histidine in it, and that histidine is mutated to a glutamine in one of the familial Parkinson's mutants. And so it could be that that residue is part of the pH dependence profile, but it may not be working independently of the other residues.
So the short answer is it's complex.
The short answer is it's complex, and P1 has no titratable residues in it, so it's not that. P2 has a single histidine in, so I suppose what we could do is mutate out that histidine and have a look. What's going on. We haven't done those experiments, but Sabina should be listening so she can write down all good ideas when we get back into our lab and can actually do something useful again.
Very good. Thank you.
Thanks.
The next question is from Vas. Do you want to ask your question directly?
Oh, thank you. I actually don't work in the Alzheimer's field, but I just joined because of like the silver lining with Corona. So I was just wondering if you could comment on why the treatments focusing on these aggregates, they've failed. I know that they've been tried on in Alzheimer's. So I was just wondering if you could comment on why they failed and why is this approach still promising? Thanks.
Okay, so I think you have to be careful about trying to think about amyloid as a generic problem, in that there are 50 known human amyloid disorders, and whilst they all have common features like they aggregate via oligomers to form cross beta, disease mechanism is a whole new slew of complexity. Now, in the case of Alzheimer's, it could well be because many of the antibodies have been targeted against A-beta, and yet tau plays a very important role in Alzheimer's disease. And it could also be because the cohort of patients, it has been suboptimal. So by the time you're showing symptoms, it's very hard to slow down aggregation and disease if you think about the aggregation kinetics with the lag phase and then the fast elongation phase. So for disorders like beta-2 microglobulin amyloidosis, there is an opportunity here because this disease occurs in all patients undergoing long-term dialysis. And so we know which people are going to potentially suffer from dialysis amyloidosis because it's linked to renal failure. So it may well be for those kind of patients that, given that they're on renal replacement therapy by dialysis already, the opportunities for, and the disease is extracellular, not intracellular, that there's more opportunities for intervention in those kind of extracellular disorders than the intracellular amyloids, like some of the, like Parkinson's and tau.
Okay, the next question is from Tarak Karmakar.
Yes. May I?
Yes.
Hi.
Okay. Hi, Sheena. Fantastic.
Hiya.
Thanks for the fantastic talk.
Thank you.
I'm a computational chemist, so my question is a little on the molecular level thing. So I was wondering if the cis2 trans conformational change is triggering the aggregation process
could stabilizing the monomer in its cis conformation by, let's say, any kind of small peptide be a good strategy to avoid the aggregation? What do you think? I mean,
yeah, I mean, it's certainly a very feasible idea. It's been shown on many. It makes thermodynamic sense, doesn't it, that if you stabilize the native state, you will disfavor the aggregation reaction. So making peptides or nanobodies that bind to the native state are indeed, as Chris Dobson and others have shown, very good strategies, potentially at least in a test tube, for preventing aggregation. And so a peptide mimic may be taken from the MHC heavy chain, could be in principle a strategy that would work. We tried that in the very early days and actually were spectacularly unsuccessful, but I'm sure that 20 years later we probably could do a better computational design. So absolutely, it's a very feasible idea, and go away and try it.
Okay, thanks a lot.
And our next question is from Samuel Kirk.
Yes, hello. Really great talk. Really nice structure stuff. Thank you. My question is about secondary nucleation. So, so you found that these interactions are highly specific. Can you comment on the interaction between the monomer and the fibril surface, or maybe some other oligomer aggregate with the fibril surface, and what might be the main structure there?
So isn't that an important question? So, so those of you that may not be amyloid experts, we know that you can get catalysis, if you like, of fibril formation by surface-mediated interactions of the monomer with the fibril surface. And that raises a really important question of what fibrils result from secondary nucleation on the fibril surface. How does it change the reaction mechanism? How does it change the fibrils formed? We've not looked at that for B2 microglobulin and for synuclein. We have done some work on that for a beta and have some nice work we're just trying to write up at the moment on that for amylin involved with type 2 diabetes. So I can't give you any specific answer about how binding to a fibril surface would change the conformation of the monomer. Although I suppose to write it down to do the experiment when we're back in the lab, some of those NMR CEST type DEST experiments are ideally suited to look at conformational changes on fibril binding. The only work we have done that you may be interested in, we published what I hope is a nice paper in JACS with Jean Baum last year, where we looked at why B2 microglobulin amyloid deposits in collagenous-rich joints. And so we looked at the conformational changes of B2M when it binds to a collagen surface. And actually what we showed there was that when the wild-type protein binds to collagen, it flips it into a more I-trans or delta N6-like conformation, possibly explaining the specificity of deposition of those fibrils. So monomer environment, whether it's a membrane or collagen or a fibril itself, that's going to be a really important part of this field going forwards.
Yeah, thanks a lot. Yeah.
Then the next question is from Trishit Banerjee.
Thank you so much. Thank you so much, Professor Radford, for this great talk. I'm actually a graduate student and I'm very new to this topic. So my question is very elementary. I wanted to know that you spoke about how N6 and P1 are so important in both beta microglobin and the alpha, the protein. So I wanted to know if there is any stark similarities or differences between these sequences, and is it something which we observe in other amyloid precursors?
That's a very important question. So well done, spot on. So I think that was what I wanted you all to start thinking about, and maybe you could give us ideas of things we could do next. So we learned from these two very different systems that regions out with the most aggregation prone regions are very important in controlling aggregation, and that the reactions, at least for these two proteins, appear to be much more specific than I had ever imagined. And I think that's where we are now in the field. And I think the next questions are, can we use these kind of methods and approaches that we and others have used, computational approaches as well, to ask specifically the question that you just posed of me is, how common are these features to other amyloidogenic proteins? And the answer to your question is we don't yet know.
Okay, thank you so much.
Okay, now Sandhya Bhatia says further down that the question is satisfied with the previous answers. So the next question would then be from Sarah Shamus. Sarah, are you still there?
Sarah's gone for tea.
Sarah says she's got a noisy daughter in the background, so please read.
Yeah, I have. Sorry, she's not quite so loud now. I've just turned the mic back on. It was such a lovely talk. Thank you, Sheena. Really well presented. So I was wondering, in P1, how well conserved between species are these 7 amino acids? Because it seems like not very many, doesn't it? And I just wondered if it was possible that there was some way of maintaining the function of the region without having this aggregation prone region in there. Do you see what I mean?
Yes, no, good question. So the N-terminal region of synuclein is not very well conserved in general. It's conserved in its features, but not necessarily in its identity. And actually, Sabina Ulamek is now actually doing an alanine scan, for example, through the P1 region and comparing the regions in different homologs precisely to answer that question that you've asked. So how do we relate aggregation potential with function and the amino acid identity of that sequence? So for example, in gamma-synuclein that doesn't aggregate and doesn't form aggregates, at least in Parkinson's disease, I don't know if Sabina's online and can answer, but I don't know how, and I can't pull up the sequence fast enough, how many residues are conserved in both P1 and P2. It's all but 2 in P1 and I can't remember P2, but Sabina, can you answer that? If you're still here, if you've gone for coffee?
Hi, yes.
Hiya. What's the answer, Sabina?
So there are 2 residues which are the same, which are different between the alpha and gamma synuclein in P1, and in P2 there are 4 different residues. No, 5. Sorry, 5.
And in synucleins in general, this is great when you can talk to your own PhD student, isn't it? And in synucleins across all of the sort of family and different species, different organisms, have you looked at that, Sabina?
Yeah, so in general, the N-terminal region is more conserved than the NAC and the C-terminal region, but it's not that the P1 or P2 region are more conserved than the rest of the N-terminal region, so there's no significant difference between it.
Brilliant. So this is a great example where the PhD student knows everything. Thanks, Serena. And thanks for not going for tea when I needed you.
Yeah, thank you both.
And so the last question that I see here in the chat is from Lewis. Would you like to pose your question?
Hey, Professor Radford, thank you very much for your nice talk. My question is totally unrelated from the analyze that you were measuring. It's more about the technique that you used.
Yep.
So I don't know if I missed it, but did you do single molecule type of experiment or assemble type of experiment?
So to get the structural information, we mostly used NMR spectroscopy because NMR gives us all atom information and is very good for looking at very dynamic systems. The only single molecule experiment I didn't do, that Kieran did with David Brockwell, was the dynamic force spectroscopy I mentioned to look at those early protein interactions in alpha-synuclein.
Okay, thank you. And then the question is, what is there ultimately limiting your time resolution when you do NMR on these samples?
So when we do the NMR experiments, we are posing the question about which species. It's a pseudo steady state experiment. So we're asking the question, which species are populated for a defined period of time at steady state, which we balance by changing the protein concentration. So we're not actually measuring amyloid fibril formation, we're measuring the dynamic excursion between oligomers present in the lag time, if you like, at pseudo steady state.
Okay, thank you.
You're welcome.
Sheena, can I also ask a question?
Yeah.
We're a little over time, but I hope you still have a minute. I was very excited by your AFM experiments, which are
they're not
fabulous.
mine, they're Kieran and David's, and I hope one of them
the
might be listening. But yeah.
And I was wondering, have you seen cases where these types of patterns look very different, where you don't have these specific kinds of interactions, and it really looks like it's all broad and smeared out and non-specific?
I don't, is David online, or has he gone to look after his kids? He's obviously not here. It's very difficult. You can't, yes, we have found situations where we don't see interactions, Ben. Of course, then it's very difficult if you don't see anything. Why aren't you seeing it? So the best experiments where you can sort of tune in and tune out interactions, for example, by changing salt or pH, as you'd well be familiar with. So you can then, in the same experiment, change solution conditions and see an interaction and lose it. So you can certainly do, we've certainly seen examples where the probability of an interaction or the strength of an interaction depends on the solution conditions. We've certainly seen examples, or David's seen examples, where we don't see interaction hotspots. Again, you have to be, we're a bit cautious about that, because how do you know whether your protein hasn't then denatured on the tip, or there's other experimental reasons that you're seeing a Himalayas of interactions rather than a specific site. So there's certainly examples of getting those kind of no hits or broad hits, but you really need careful controls to show that it's a real result and not an experimental artifact.
Yeah.
But I mean, some of your IDPs would be great to try.
Yes, no, I was thinking of,
yeah,
that would be very interesting.
So maybe we could talk offline about doing that. That
would be great. Yeah, so with that, thank you very much, Sheena, for this beautifully detailed, structured views of amyloid formation, and really going from single molecule experiments all the way to worms. Very impressive, beautiful work. And many people in the chat commented on your beautiful lecture.
Well, if anyone has any other questions, we're in lockdown, so I might even answer an email.
And I would also like to thank all of you for joining us today. And I hope that I will see you again in 2 weeks. And let me just briefly share my screen to bring up our website, which I hope you can see now. And so we had Sheena today, and in 2 weeks, we will have a lecture by Ken Dill. And I can promise that we will have more lectures in the future, and which we are currently setting up. And so please have a look at the website if you would like to know what's going to happen next and which lectures are coming up. Also have a look at our YouTube channel. So Jane's talk is already here on the YouTube channel. Sheena's lecture will be there shortly. And so with that, thank you very much again for joining. Thank you very much, Sheena, for a wonderful talk. Hope to see you again in 2 weeks. Stay safe, take care. And I hope that more and more of you can go back to the lab. And so we will be able to show more and more interesting results about protein folding and dynamics. Thank you very much.